Oh, hello there. You came just in time. Today, the lab received an exceptionally preserved sample of a bone from Tyrannosaurus rex, a dinosaur that lived 68 million years ago. With the right techniques and a little bit of luck, we can learn about the biochemistry of this ancient beast. Let's go. The first step is to demineralize the bone in an EDTA buffer for 7 days. This will get rid of the inorganics and leave the biomolecules behind. While we wait, let's talk about what we can expect to find.
First of all, you can forget about recovering DNA from this sample. Even at cold temperatures, DNA is thought to degrade too quickly to last 68 million years. For example, this graph shows that a 100 base paired piece of DNA at 15 degrees Celsius has a half-life of only 1,000 years. It's no wonder that the oldest DNA ever sequenced was less than 1 million years old. Protein, on the other hand, stands a fighting chance of lasting 68 million years, especially if the conditions are right. The graph on the right shows the immensely lengthened lifetime of protein when it is closely associated with bone mineral. This is exactly the environment of the T-Rex protein. However, even if we recover protein from the T-Rex bone, it may be badly degraded. One degradation pathway of particular interest is the deamidation of glutamine to glutamic acid. This process occurs slowly and predictably enough for it to be used to estimate the age of ancient proteins like those in the T-Rex. Overall, we know surprisingly little about how proteins naturally decompose over time.
It's been a week, and the bone of our Tyrannosaurus rex has almost completely demineralized. Collagen 1 makes up 80% of the protein in human bone, so my hypothesis is that the T-Rex protein is also collagen 1. I will divide up the protein sample and test this hypothesis in several ways.
First, let's examine the overall amino acid content of this T-Rex protein. Collagen 1 from chicken bone is known to have a glycine to alanine ratio of 2.5 to 1. Data from time of flight secondary ion mass spectrometry indicates that this T-Rex protein has a glycine to alanine ratio of 2.6 to 1. Not bad. Second, modern collagen is known to contain the amino acid 4-hydroxyproline. This post-translational modification of proline is needed to stabilize collagen's triple helical conformation. Hey look, mass spectrometry spotted this residue too. Finally, given the amount of structure conservation that's been observed for collagen across the animal kingdom, it's reasonable to expect T-Rex collagen to have a tertiary structure similar to that of chicken collagen. An immunochemistry experiment confirmed that this was the case. As seen on the right panel, the antibody to chicken collagen also has a strong binding affinity to T-Rex collagen.
Now that we're confident in the similarities between T-Rex collagen and chicken collagen, we can try to investigate the differences between these two proteins. We can do this by chopping up the T-Rex protein with trypsin and then using mass spectrometry and various computational tools.
Mass spectrometry tends to perform poorly when it attempts to infer peptide sequences from their spectra alone. Direct de novo sequence identification is even harder when the quantity and quality of protein is low, as is the case for the T-Rex collagen. Reference peptide spectra and known protein sequences often support the mass spec data to provide more accurate peptide sequences. One such algorithm that combines mass spec data with known sequences is BICEPS, Bayesian Information Criterion-Driven Error-Tolerant Peptide Search. Thanks to its sophisticated workflow, BICEPS outperforms many of the algorithms that came before it. As mass spec technology improves and known sequence databases expand, we can expect to get even more accurate peptide sequences. As technologies for analyzing ancient proteins continue to advance, scientists may start to investigate how dinosaurs used biochemistry to their advantage. For example, this chart shows that T-Rex was even heavier than an elephant and that Brachiosaurus was heavier than both combined. Did dinosaur bone collagen adapt to support such tremendous weight? Future research of ancient proteins may give us the answer. Sam Allon, the Science Mon.